Good afternoon, everybody. You're very punctual, far more punctual than I'm used to in medical students when they usually turn up 5 minutes late. So congratulations. So I'm Adrian Harris. I'm Professor of Medical Oncology here in Oxford, and I run clinics to treat patients with breast cancer, but head of a group of doctors that specialise in cancer therapy, part of a much bigger team working in the health service of 50 consultants in medical and radiation oncology. And one of the big advances, in very simple terms, is working as a team, the team effort of treating cancer of every type, involving surgeons, medical oncologists using the chemotherapy and new drugs, radiation oncologists for radiotherapy, immunologists now, and imaging. So those team efforts have made a massive difference over the years. I've actually been training as a doctor for nearly half a century since I was a medical student, and it's just amazing to see what's happened. So let me start off by saying also no medical student sits on the front row, and that's because they're worried about being asked questions. I guarantee I won't ask anyone a question who's on the front row, but I might ask some of these people sitting at the back unnecessarily, okay? So the first question is, back there, does anybody know what this molecule is? Any ideas what that molecule might be, apart from the fact that we've tricked the word cancer into it? Very good. Top marks, DNA, yes. So everyone knows now that DNA is the molecule of life. And just to give you the background story of cancer, there's 33,300 cases per year in the United Kingdom. And half of those patients will die from their cancer, but half will be cured. And the first time last year we could say, this century, that half of all patients with cancer will not die of their cancer. And so that's an amazing step forward, actually. 1 in 3 people will get cancer, half will be cured, and of all the deaths that occur in the UK, 1 in 4 are due to cancer. But the very important message I'd like you all to take home, regardless of fantastic breakthroughs, technology, 40% of all deaths from cancer are preventable. I'm going to ask our audience to shout out any preventable causes of cancer. Smoking, yeah, that's number 1, and that will probably reduce all cancer by 30%. Any others? Alcohol? Too much sun? Very good one. Melanoma's going up. Any others? Yes? Sorry, yeah. Papillomavirus causing head and neck cancer and cancer of the cervix. Yes? Any others? Sorry? Exercise causes cancer? No. It's No, yeah, that's right. No, it's okay. Yeah, but you're absolutely right. Lifestyle exercise does prevent cancer, and it also can improve the survival of people who've had cancer. So very important. So I think you all know about this, but probably most of you, apart from the smoking, aren't taking too much notice of it. But nevertheless, diet may also be important, but more related to obesity than saying one particular thing, apart from probably red meat and colon cancer. So all the others not really proven, apart from red meat can be associated with colon cancer. The others are sensible lifestyle choices because they affect heart disease as well. So 40% of cancer is preventable, and we must try and improve on that. And of course, one of the things I used to treat lung cancer, everyone I've ever met had lung cancer said their grandfather lived to 80 and smoked 20 or 40 a day. And it's not ridiculous, because some people will be able to take very life-threatening approaches to diet, smoking, alcohol, and survive. So that tells you right away there are genetic differences between people, so our risks are not all the same. And that will be one of the steps forward, is trying to work out what people's risks are, to know who should make which modification. And that's a lot of the work that's going on in the epidemiology department, where it's now got molecular, so what are the pathways that make some people risky and some not? But we can't bring that to the general public yet, but it will be in the future. So now I want to turn to what are the common cancers. I've just put the 4 most common cancers in men and women, which account for about 50% of the cancers. And what you can see in men, it's prostate cancer, lung cancer, bowel and bladder. And lung and bladder are both smoking related. Breast cancer in women, the commonest, then lung cancer, smoking related, so very sad to see that. Bowel cancer and uterine cancer. But when you can look at the causes of death, the causes of death are not the same as the ones that are common, because we've made quite big inroads against the common cancers, so people are not dying from them, including screening from colon cancer, breast cancer, cervical cancer. You see that the causes of male death are lung cancer and prostate, bowel cancer, but rather than number 4 here, esophageal cancer, which is also related probably to a virus infection and is increasing in incidence, and we don't know why yet. In females, you can see although breast cancer is the commonest, the death rate's much lower, whereas lung cancer is now the commonest cancer cause of death in women. Entirely preventable, or nearly 90% of that is entirely preventable, 10% is not due to smoking, it's bad luck. And, but so very sad to see that this has risen over the last decade at the very time that we're beating breast cancer. And you also see pancreas cancer, another rare cancer, very hard to treat, is creeping up as a cause of death. So if you look overall, we are winning the battle against cancer. This looks at the since 1975, 85, 95. That's when I qualified for medical school, 1973. And you can see that the death rate amongst women, it was pretty steady, and then a gradual decline from 1990, just going down. Same for men, and also the two average across the two. So what's more caused this really clear-cut trend, which is across Europe and the developed world? And it's not just one thing. A lot of it is prevention, screening, and then doing what we know, doing it best as a team. And that's been very important. That's the way it all works here now. But without the basic discoveries of the last few decades, of what you're going to summarize, how they've been made and how they link to our new treatments, we wouldn't be obtaining such a big effect. So I've got here imaging, because we can see tumours much better to see whether treatment's working or not. We can understand what makes a tumour grow now. Surgery is massively better. And we have many new drugs and new ways of giving radiation, which are far less harmful and more effective. So how does cancer develop? What gives us cancer? Well, you'll all recognize this DNA molecule you've already seen. And these little pairs of bases are a genetic code, which I won't go into detail, but every cell in the body has 3 billion of these little base pairs. Okay? 3 billion in one cell, and there's 13 billion cells in the body. And so you can see how much complex information. It's round little spools called histones that pack into fibers that pack into our chromosomes, and we have 23 pairs, one from each parent. And so you can see this must be a big job. If one of those goes wrong in the wrong protein, you can get cancer. So how can we possibly manage not to get cancer, you might say, when you've got to make, every time a cell grows, you've got to copy 3 billion base pairs accurately to make a second cell. Amazing problem. Now, I've showed you the length of the DNA. That makes about 30,000 proteins. Like, you obviously got hair proteins and lens proteins and hemoglobin. There's actually 30,000 different ones that make up the different cells of the body. So how do you pack all this in? Has anyone got any idea, if you unravel this DNA that's packed in with 3 billion in every cell, 13 billion cells, how long would that DNA be if I put it all end to end from every cell in your body? Any idea? That's a good start. Anybody above 100 meters? How much? 180 meters. Anybody higher? How about a mile? Do you think that's possible? 10 miles? Possible? 100 miles? Possible? 1 million miles? 93 million miles, more than that. Your DNA, stretched end to end, would go from here to the sun and back several times. That's unbelievable, isn't it? But I can do the sums for you later. But when you realize the size of that per cell and how many cells you've got, and it shows you how complex our DNA is, which is our genetic stability. So it's hardly surprising things go wrong, and it's amazing we can live to ripe old age. So how does it go wrong? Well, first of all, every time a cell grows to replace a worn-out cell, it's got to double it. So the DNA and these little bases on it can get abnormal, it can get a break, or it can get damaged by free radicals, or it can link across wrongly. So all these things are sort of built up in your DNA. So I mentioned there's 3 billion in every cell, and 10,000 a day in every cell are being damaged. So you've just got, in every cell of your body is getting 10,000 hits a day in every cell that are damaging it. So you can imagine evolution has had to protect it. So we've got brilliant ways of repairing DNA, which is one reason why we live so long, because we can repair that damage. But nevertheless, every so often it's going to get through. And as you get older, you're going to get more damage accumulating. And so what happens is, this damage might be somewhere harmless. It might be in a hair cell. It won't matter. But if it is in a gene that controls how cells grow, it may switch it on all the time, so it doesn't switch off. When you cut yourself, the wound stops growing after a while, doesn't it? It just heals. But what switches it off? There's a special switches that do that. If you get a mutation, damage to one of those switches, it won't switch off. And so common areas that get damaged include the line of the mouth, the skin, the lung. And this is happening all the time, but we're really good at repairing. Now, some genetic defects, like breast cancer genes, can cause defective in repair and give an increased risk for that, for example. So, bearing this in mind, our bodies have evolved to protect us from all this, and they've got fantastic protection. There's 10 milligrams of well-known carcinogens into burnt toast, the burnt bit. And our body, we don't count toast as something dangerous because our body is so good at getting rid of those carcinogens. But if you paint them on skin of mice or expose bacteria to them, they get mutated. So that's the first thing, is we've got damage all the time and it's being repaired. Secondly is, what switches these genes on? I told you 30,000 genes. They're not all in your eye or all in your muscle, in your heart or your lung, are they? Every tissue is a bit different. So there must be little switches that turn on the genes and off. So you've got one pattern for cancer, for lung, one cancer for skin, one cancer for heart muscle. So all these little switches are there switching on and off as well. And I'm just giving you a little technical problem because there's probably about 1,000 proteins that make these switches up, scattered on all these genes. If you look at Lego, there's the classic Lego brick, 8 pins. If you take 6 bricks with 8 pins, how many different ways are there of joining them up? Any guesses? I'll give you some pretty big number before. 100, 1,000, 10,000. Any guesses? Millions. Who said millions? 915 million. Well done. No prizes though. So what I'm simply saying to you is complicated, and these switches don't have just 2 bits of Lego. They've got 20 or 30 or 100 bits of Lego scattered around to switch on and off. And these can go wrong as well through the same effects. If they get damage to the DNA that makes them. But because we now know through gene sequencing what's happened, we can actually make drugs that will stick and Lego block the ones that have gone wrong so that we can correct the defect. And there are new drugs in the clinic now that are looking really effective for that. So that's by understanding the science of what switches them on or off and making molecules that will block those abnormal ones, we are now starting to do that. And that some of the trials we're doing in Oxford are on these sort of drugs. So that was how we switch genes on and off. But the next thing is what makes cells grow. And I mentioned before, wound healing. When you cut yourself, substances called growth factors are switched on that make cells grow. So, as I said to you before, the normal DNA is 3 billion base pairs in each cell. And if DNA damage, like ultraviolet light from sunbathing, chemicals from smoking, cause a mutation, that's a bad hit in an important gene, then becomes called an oncogene, and it can make cells grow. So this basically, one of your own genes has become abnormal by being damaged, as I've described to you already in the DNA, and it then makes an abnormal protein that's on all the time, instead of just when you need it. So that makes a cell cancerous. Now, if you know what that is through basic science discoveries in the last decade, you can make some way to stop it. Without knowledge, you can do nothing. And so people discovered an important gene switched on in breast cancer on the surface of cancer cells called HER2, and that these growth factors activated it and made the cancers grow very quickly in breast cancer. So they made an antibody to block it, so they stopped it signaling inside the cell. And this is looking at patients who've got advanced breast cancer spread around the body, and this is a group of patients treated with a standard treatment, and this group of patients had an antibody to block that added. And you can see that even with recurrent breast cancer, you can survive for several years. But that the patients who didn't get the new antibody, got the standard treatment, average survival is 40 months, and average survival is 60 months or more with the antibody. So a single antibody for advanced cancer can halt it at least and improve it. And that's through the discovery of this particular oncogene. And there's many others in development. So antibody therapy is another major advance, common in many types of cancer, but particularly breast cancer and lymphomas and leukemias. Now, another major breakthrough in the last couple of years has been in melanoma, which used to be pretty well fatal if you'd spread around the body within 3 months in general. Now, I mentioned to you these switches. What switches the cancers on? Showed you about the HER2 in breast cancer on the cell surface. But deeper inside, there's other signals, and one is known as BRAF. And this is a scan, and I mentioned to you imaging. So this is a scan of a patient using a PET scan, which measures how much sugar, glucose, cancers take up, because they use a lot of glucose. And you can see this patient, poor man, is riddled with cancer from his melanoma. But because they knew that there was a particular gene, a particular switch which was on, and a particular damage to it, they had found a drug that could block that, was made specifically for that. So this drug could stick only to the abnormal protein, not to your normal ones, so it wouldn't cause any normal tissue damage, only to the one abnormal type protein, and this is what happened 2 weeks later. You see? Amazing. So that's what's called targeted therapy. So that was an example of one gene in melanoma that was wrong and blocking it. Now we know there's over 400 different genes that can go wrong, and people are now there's no drugs to block each of those in turn. But you can see there's no good giving a person a drug if you don't know what's wrong with them. So this isn't the only thing that causes melanoma, and so you have to do tests on the patient's tumour, or now even in their blood, because the tumour leaks DNA into the blood, so you can see what's wrong with the tumour from a blood sample. Another major breakthrough. We can see which patient should get which drug. So this is forefront. It's happening now in trials, and it's in standard practice for some cancers already, for lung cancer, which is not due to smoking, for example. So we made major breakthroughs. We understand what causes cancer, what drives the cancer, how to make drugs to block it, how to pick the right people, how to see if it's working. So this has contributed massively to the last decade, and it's a very exciting phase going forward with being able to measure this in the blood of patients and see how it changes on treatment. Now, I'm going to talk a bit about some of my own work. So cancers can't grow and spread without a new blood supply. So they can grow to about 1 millimetre in size, with lots of green cells there, but they need the blue blood supply to give them oxygen, to help them grow, and also once that happens, they can spread around the body and repeat these other processes. So this is a very important early step. So one of the things you should all know is blood from where you don't expect it is a sign that you should go to the doctor about. So postmenopausal bleeding, blood in the urine, blood in the stool, blood coming out of your ear, blood out of your nose even, unless you've blown it or someone's punched you, blood coming out of your mouth, bleeding gums, you should see your dentist. And one reason why this is the case is that when the tumour's beginning to grow, then it needs these new blood vessels which are growing much more quickly than normal and are very fragile, which is why you get that. So it's a very old sign that everyone should know about. It's got a very good scientific basis for it. It's not just luck or bad luck. So I'm very interested in what happens there, because we showed for the first time a long time ago now that blood vessels in cancer are growing 50 times more quickly than the blood vessel in normal tissues. So if you could try and block the blood vessels in cancer, you might have a general treatment that would go around the body for many sorts of cancer. And one of the molecules we worked on was this little green molecule I've shown here. There's a cancer, and there's little green molecules going to a blood vessel nearby, and there's the lining of the blood vessel and the blood inside it. And as the tumour grows, it makes these little green molecules that bring a new blood supply to the tumour, which then allows it grow. Now that little green molecule, we can see close up. It looks more like this. That's the surface of the blood vessel. And inside the blood vessel, when the green molecule sticks to the red one called vascular endothelial growth factor receptor, it sends a signal that makes the blood vessels grow. So if we could stop that, we might be able to stop cancer growing. Now, is this real? I'm just showing you a cartoon. This is a picture from breast cancer. It's a sort of breast cancer which is non-invasive, early, and you hope to diagnose by screening, so 98% cured. And what you can see here, this is the tumour, and normally you should have a hollow duct for carrying milk, but this is full of tumour now, and there's a blood vessel going along, and it's been hijacked, and it surrounds that tumour. So that's going to go off, but of course we moved it before that happened, and that's what an normal blood vessel looks like. And you can see how this has been hijacked. So we can see it really happens on a real slide of a tumour. Now this is another slide of a patient who's got kidney cancer, and it's spread to the skin. And kidney cancer is known to produce more blood vessels than any other sort of cancer. You can see, even though the cancer is under the skin, it's leaking out this VEGF, and it's causing this massive angiogenesis. So what can we do about it? Well, we've actually, many companies now made drugs that will block VEGF, this angiogenic factor, there's an antibody binding it that will stop it stimulating the blood vessel. And there's 10 cancers now where that is part of the standard treatment, so it's become routine practice to use this as part of the standard treatment, not on its own, but working with other mechanisms. I hope I woke a few people up there. But that was to show the effect of blocking VEGF. Now, of course, this doesn't really happen in patients, otherwise you'd hear a lot of noise when you came to clinic, but that shows the general principle. Now, one of the things I think I've shown already is one size does not fit all. You need to know who it's working in, and this is where the imaging comes in. So what we can see here is a breast cancer. This is, I work mainly on breast cancer. And we can see here, this drug that I just showed you the cartoon of blocks VEGF. And this scan is a special type of scan called a magnetic resonance imaging scan. And you can see the blood supply around the edge of the tumour. And what's happened in this particular patient, you can see the blood supply around the edge. And it's gone down very markedly after just 2 weeks. This another patient, there's the blood supply. And in 2 weeks, the centre of the tumour's died. So a single shot of a very low toxicity drug has killed 90% of the tumour for this patient. But this patient, you see the blood vessels, 2 weeks later, they're worse. So these patients all had the same drug, no previous treatment, but some did it really well, others moderate, and some even grew through it. So this means this patient hasn't got VEGF, it's got something else growing in the tumour, not VEGF. This is the result we want. And this tumour somehow is managing to survive without the blood supply. So this has helped direct our research to new pathways and mechanisms. So we recently discovered a new mechanism for the blood vessels to grow, which we're now working on. We're trying to block it with antibodies. And survival pathways, which seem to be somewhat similar to what happens in metabolism in diabetes, and metformin may help. A drug used to treat diabetes may help that sort of cancer. Now, it's going to switch now from breast cancer to kidney cancer, which I already showed you had that massive blood supply. And again, we have drugs that target the blood supply. Can anyone, I think you can see here, this is a CT scan, a CAT scan of someone lying on the floor. That's their back. Their feet are sticking out. And there you can see in the ribs a great big lump compared to the lump on that side, nothing. Big lump, and the blood vessels growing into it, but not quite into the centre, which has got low blood supply getting into the very centre. And a few weeks after giving a drug that blocks the signalling pathway, it's gone. Dramatic result, which lasted for 2 years. But what you can sometimes see, unfortunately, in many cases, that cancer can always try and outsmart what you do. So this is pretty smart stuff. It worked very nicely for that patient. But we can see in another patient who wasn't so lucky, the drug, and this is another CT scan, but this time it looks at the blood supply in the tumour. So there's the pelvis, the back, the feet are sticking out, and all the yellow bits are the bits of cancer in the bone from his kidney cancer. This is just dye in the gut. And so you can see those bones should be blue, and they're yellow because of the increased blood supply. And then about 4 weeks later, you can see these yellow bits have gone pale again, like they should be. You see, all that has gone pale. It's almost completely gone, right? So a very good result. But again, about 4 months later, the blood supply has come back in exactly the same area, even worse. So how did that happen? So the tumor, we've blocked one pathway, and the tumour's used another. And that's what our research is on, to try, and this is why we need our patients to help us and to help themselves as well, and other future patients. We need to get biopsies and understand what happened there. But without the scanning, we wouldn't even know that because we wouldn't be able to see that. So I think the working together of the team, the surgeons, biopsy, our patients, of course, the imaging, all comes together to try and develop improved treatments. And then a final bit I want to concentrate on is immunotherapy. In terms of systemic treatment, and you've seen a lot of excitement about this. This is a very complicated picture, and believe me, it's just the start. But we now have some very exciting results based on this science, which is the science led the discovery of these important new antibodies, which are curing patients with advanced cancer, with melanoma. A third of all patients are probably cured forever by this antibody treatment. Bladder cancer, lung cancer. So a really amazing, radical change in how cancer is treated. And so this is a tumour cell in grey. And maybe you've heard of antigens. These are proteins that cells have on the surface that are different from normal. And the cancer cell, because it's got these abnormal proteins, the mutant proteins I told you about, the switches that are changed, they will have different antigens on the surface than normal. So we have cells in the immune system, which you've probably seen about already on the presentations in the other room, T cells that kill them. So T cells spot this foreign antigen the tumour cell is making because it's got these different proteins now and tries to kill it. But tumour cells are smart and they make a molecule that switches the T cell off. So the T cell tries to kill it because it recognizes that. The tumour cell makes PD-L1, which tells the T cell, stop killing me. And so that's why many people don't have an immune response anymore, because the tumour's actually told the immune response to go away. But if you then give an antibody that can block the tumour sending that signal, or block the T cell receiving it, then the T cell's killing that cell, but the cell can't say switch off. And these are the pictures of some of the dramatic results in melanoma. Very early studies of 4 years ago now, and they show that in this group of patients, this is up to 2 years now, that we know up to nearly 10 years, that a group of patients, within a few weeks, the tumours have shrunk and stayed away. But others, it carried on growing. So once again, we see about a third of patients probably benefit extremely well, but the others not. And why is that? So if we didn't know the science, we wouldn't be able to work it out. But now we're starting to work out what it is about those other patients that stops that drug working and coming up with other drugs. So you can see it's a continuous evolution. No one size will ever fit all. Cancer will be cured and is being cured already, but it's by getting the right team with the right drugs and understanding that particular patient's tumour and why that responds and why that doesn't. And I think Oxford is at one of the forefront of trying to look at this across many diseases. And this is just a picture from one of those patients. In fact, Oxford is one of the leading centres for melanoma research. You see all these blobs. This is again on their back, feet sticking out, ribs, and big, massive. See how big some of these lumps of tumour are scattered around and how they've shrunk within about a month or so. So we never saw this before. So it's really amazing how the science has helped to understand immunology, make the drugs, and treat our patients. And then those are all systemic treatments. They go around the body. They help people around the body, everywhere. But probably 1 in 5 patients don't die because of the cancer spread, but they die because the cancers come back in the area where it first started, and head and neck cancer, lung cancer, cervical cancer. So being able to treat where it started very well is important, or if it comes back is very important. And traditionally we've used X-rays for this, and this, of course, the X-ray beam is colourless, but this just shows the dose given to someone who's got a lymphoma, curable disease, and there's their arms raised above their heads, there's their jaw and face, their ribs, and they've got glands here in the chest and the armpit that have to be treated. So this is the standard way. See, the red gives the full dose, but there's a lot of spread of lower doses around the rest of the arm, the neck, and the chest, which is not good for the tissues. But and then you can see when you try and looking at it from above, you can see the beams going through the arm, and there's a beam going to the lymph glands behind the sternum, the chest breastbone, but you can't avoid hitting the heart. But with a new sort of beam that's being built in Oxford, the proton beam therapy, it's much sharper, much more concentrated, so you can actually get it in a much better way to where you need it. And this here shows what happens with proton beams. You can see this big area of blue that's going around the rest of the tissues is missing now because it's much more accurate. And it also stops, and you can stop it going too deep. So you can see it just stops at the edge of the heart instead of going all the way through. And you can see far less tissue damage rather than doing all this tissue at the back. So this is going to be another great breakthrough for Oxford to have this sort of therapy available to link to both systemic treatments and local treatments. That's where it started. And finally, we mustn't forget the surgeons who cut out cancers and are major contributors to the cure of cancer. And this is a new technique being developed by Freddie Hamdi, Professor of Surgery here. And this is using a special dye, a fluorescent dye that shines under ultraviolet light. And it's taken up by cancer cells, but not normal cells. So you inject it into the patient's body under our operation, and you look into the abdomen for your laparoscope, and just the cancer spots light up. So you can see tiny spots we could not see before, and you can be sure you removed all the cancer, which we could never do before. It's just by eye saying, does it look like that? So you've got a much more accurate way of doing it. And this medical student's doing research on it, and this is the fluorescent kit that was developed in Oxford. And it's being already used in bladder cancer, ovarian cancer, and other tumour types. Of course, we don't know yet how much better it's going to be, but it makes sense. If you can remove all the tumour and see what you're doing, you'll do less damage to normal tissues, and you'll remove more cancer. So I'd just like to point out how much money has been spent in Oxford. £125 million on the new cancer centre, state-of-the-art radiotherapy, which are the best in the country, and certainly as good as the best in Europe. This is what I first started off with when I came here 50 years ago. My first cancer ward was a Nissen hut, so I think it's a big improvement. And this is an idea of what's going on in Oxford. Hopefully none of you will have to come across this, but if you do, it'll be as good as you can possibly get. There's radiation, I mentioned, surgery, medical oncology, the basic science on the DNA, the blood vessels, the spread of cancer, the epigenetics, which is the Lego block stuff, the tumour immunology, all big groups working on it. All these research institutes, including the epidemiology, and the prevention of cancer, and special trials, early trials, the first in man, being done here. So I think, first of all, we're lucky to have patients who will volunteer and go into trials. Sometimes they're not to their own benefit because they're new drugs, we don't know if they're going to work or not, but without their contribution, none of this would be possible. So the vision of the future is actually nearly here. From a blood test, or a simple needle test from a tumour, you can extract, DNA, we can check the mutations and the abnormalities, and tells us what drugs are going to be the best for that patient. That's a special kit. Can you see that it's run by a dilithium crystal? You know that dilithium is not yet invented, but that's going to come in the future. And with that, we know what drug to give the patient. And lo and behold, we've cured the patient. Now, what you'll notice, however, unfortunately, he's got some fox-like features, and it's inevitable that we do have side effects. I'm sorry about that, but you do get side effects. So, like that, I'd just like to stop and say, hope you enjoy the talk.